The overall goal of this procedure is to illustrate a technically simple yet highly reproducible protocol for successful primary culture of human vestibular schwannoma tumors. This is accomplished by first timely processing the tumor sample with attention to sterile technique. The second step is to mince the tumor sample into sub-millimeter cubed fragments in ice-cooled conditions. Next, the tumor is further dissociated by enzymatic digestion and serial trituration. The final step is to plate the tumor suspension on pre-treated culture plates. Ultimately, immunofluorescence microscopy is used to evaluate culture purity by evaluating cell phenotype and to evaluate changes in functional outcomes such as proliferation and apoptosis.
So the main advantages of this technique over existing methods are first, the simplicity of the technique. Second, the decreased time commitment that's involved in preparing the cultures. Third, the reliability of being able to produce highly purified cultures. And fourth, the fact that the cell phenotype and behavior in vitro in these cultures mimics the behavior of the tumor in the patient.
Begin the setup by coating the cell culture plates. Working in a sterile culture hood, add enough 0.01% poly-L-ornithine solution to cover the bottom of a polystyrene culture dish. After replacing the lids, let the dishes sit at room temperature for at least 2 hours. After 2 hours, suction off the poly-L-ornithine solution and allow the plate